The first thing you need to know about biology, and any system in biology that you study, is that it's affected by evolution. So technically, evolution is defined as a change in heritable characteristics of populations over successive generations. That is, characteristics that are passed from parent to child or parent to offspring over time, right? That's what heritable means. So when we talk about heritable characteristic, what are some examples of heritable characteristics? What are the things that you can inherit from your parents? So eye color, yeah.
Height.
Height.
Certain diseases.
Right, you can be genetically susceptible to certain diseases. If we're not talking about humans, right, here's a good example. These are finches from the Galapagos Islands. You may have heard of Darwin's finches. So these populations of birds, when Darwin visited the Galapagos Islands in the mid-19th century, first kind of gave him an idea of how evolution would work, because he saw that these finches are all very closely related species. They all clearly have a common ancestor, and yet some of the finches have these big, fat, thick beaks, and some of the finches have these smaller, pointier, narrow beaks. And what he realized was the finches that lived on islands with mostly fruits and seeds had the thick beaks, because you could use more force. You could crush open the nut or seed and get at what's inside. The finches that had the longer, narrower beaks were on islands where they mostly fed on insects, and so they could reach into burrows and grab insects to feed. So the beaks had changed over time to allow them to optimize to the food source in their location, in their area. So how do those change? How do these characteristics change? Basically, two big words, right? How do heritable characteristics change over time? I'm sure you've heard these words before.
Natural selection?
Natural selection, thank you, right? These finches underwent natural selection. The ones on the seed islands that had weaker beaks couldn't crack open as many seeds. They couldn't make as many offspring. They weren't quite as healthy, so they died out. The ones with the thick beaks on the islands with the insects couldn't get their thick beaks in the hole enough. They didn't feed well, they died out. That's natural selection. Right? Almost anything that you see, any characteristic that you see in an organism was put there by natural selection. So your brain, your great big brain, was put there by natural selection, because humans that had bigger brains, that had more cognitive ability, had faster reflexes, all those things, did better in their environment than the ones that didn't. And we are all the descendants of those big-brained, fast reflex individuals. And that was evolution by natural selection. So anytime someone tells you evolution is just a theory, you tell them no. Evolution is a fact. Genes change over time. Genes mutate, you pass down to your offspring. There's always a little bit of change every time. That's not open for discussion. The method by which these genes change, that's what people argue about. The theory is called the theory of natural selection, or what Darwin calls evolution by natural selection. So when we talk about the brain and the nervous system, they're not immune to evolution by natural selection. We'll see how things changed over time through evolution. And there's a real nice quote from Dobzhansky, Theodosius Dobzhansky. Nothing in biology makes sense except in the light of evolution. So any biology class you take, from ecology to molecular cellular biology to animal physiology, you're going to hear about the effects of evolution. So in terms of the nervous system has evolved over the past 500 to 600 million years that multicellular organisms have been on this planet. And basically what you can see is over time, nervous systems have got more complex. So This little guy is a hydra. Hydra is basically a little aquatic organism. It's really just a stomach with arms. That's pretty much all it is. Anyone have a fish tank, ever have a hydra in it? You know, they sell them at the pet store. You can get them if you want. So they kind of fix into a certain place, and then their little arms bring little scraps of food to their mouth, and that's pretty much it. Pretty basic life for a hydra. Their nervous system, these guys are hundreds of millions of years old. These are ancient. Their nervous system is called a nerve net. Basically just single nerve cells that are connected to each other in a network. That's a very simple nervous system. So really it's like if you're a computer scientist, if you're interested in computers or information, right? You can connect a whole bunch of different terminals, but if there's no like central processor, there's no server to kind of mediate the flow of information, it's not going to be as useful. That's a nerve net. Very simple. It basically, you know, if you touch it, it'll move away, enough to move their arms towards their mouth. That's pretty much it. So we don't think hydras have a very rich internal life. Centralized nervous systems are a little bit more advanced. They show the beginnings of a brain, a section of the nervous system that's enlarged, that has more cells, and that does more powerful stuff. So the earthworm is an example of a centralized nervous system. So invertebrates, like earthworms, you notice they're segmented. So just like in worms, the nervous system is segmented. So if we look at an invertebrate nervous system, right, the animal has segments to its body, and each segment has a little cluster of nerve cells called a ganglion. But up towards the head of the animal is a slightly bigger ganglion. It's got more nerve cells, it does more stuff. That's what's called a centralized nervous system. Not really a brain yet. It doesn't do all the functions that we would expect a brain to do, but it's a step. It's an evolutionary intermediate. Finally, when we get to vertebrates, right, very highly, well, I don't like to say highly evolved, but that, right, hydras and earthworms are just fine, too. Evolution doesn't care. You know, all of these three animals, evolution doesn't hold one above the other, so. But we are more complex by any means than, say, earthworms or hydras. So more complex animals, like vertebrates, that includes fish, amphibians, reptiles, birds, and mammals, anything with a backbone. We have a cephalized nervous system, which means we do have a very large, concentrated group of neurons, which are the brain. Any vertebrate you can look at has a brain, and that's called cephalization. When your centralized head ganglion gets big enough, we call it brain. So those are really the three steps of nervous system evolution. We went from a bunch of just interconnected random nerve cells to better connections with maybe a slightly larger one at the front to what we consider a brain, right, which is the very complex processor. Now you'll notice that all of these brains, whether it's a larger ganglion or a true brain, are at the front of the animal. Why is our brain in our head? Right, that's an interesting evolutionary question. Why is our brain in our head? If we think about it as a product of evolution, right, we should expect that there should be some advantage to having a brain in your head, right? Because otherwise, there wouldn't be any natural selection towards having a brain. And since every organism, every vertebrate on this planet has a brain in their head, there's clearly got to be some advantage. So what do you think an advantage might be to having your brain in your head? Or at the front of your head? What do you think?
Is it that all of the nerves in the body stem from one point and just go in one direction?
That's true, but it's like, for example, right, for humans it's down, but it's not always down, right? from a fish, it would be laterally. So yeah, I mean, they do.
I just don't know if having one direction was an advantage compared to having it like branch off in different directions.
Well, there are a couple ways we could think about that, right? Yeah, so if you have everything centralized, right, in one location, you have one brain. So yeah, if you had more than one brain, right, they might conflict with each other at times. So it's always good to have one brain. But why in your head? Why not in your left butt cheek?
Is it because it's protected by, like in these humans, like the skull?
So yeah, right, so you protected it. Although they've evolved together, right? The brain has evolved and the skull has evolved to protect it. But yes, you do have protection from your skull. What else?
It's close to your lungs, so like you can get oxygen in your heart. I think, I don't know.
It is, but it's, you could, your circulatory system and your respiratory system are so robust that it could pretty much be anywhere and get plenty of oxygen. What do you do with your head?
Look and stuff.
Right, you lead with your head, right? Your head's the first thing that's going to encounter any strange environment. If you're not sure if there's an axe murderer outside the door, what are you going to do? You're not going to put your foot out first, or maybe you do, I don't know. You're going to peek out here with your head. So basically, the brain's in your head because that's where you lead with. That's where your sensory organs are concentrated, right? We have eyes in our head, ears in our head, nose in our head. There's not much reason why we have to do that. We could have eyeballs at the ends of our fingers, but we don't, because the less distance the information has to travel, the faster it gets there. So we have all of our sensory organs clustered in our head. And we lead with our head. So the first thing to check out some new environment is the part that has all the thinking. Right? And that's another advantage, right, is having all the nerves clustered in one place. It's like having a computer with more RAM or a faster processor, right? The more nerves you have in one place, the more cognitive ability you have, the better thinker you are. And as we can see, right, out in nature, animals that are the best thinkers generally do the best in terms of survival, right? Humans are the best thinking animals. We are all over the world. We are far more widespread than any other given species. And part of it is because of our big brain and our ability to have what we call higher cognition. Right? So clustering your sense organs around your brain, having a big brain that gives you much better cognitive abilities is a very good product of evolution, a good reason why your brain wound up in your head. Now, there are a couple of disadvantages, right? Kind of touched on some already. What's a disadvantage of having all the nerves in one place? What's a potential drawback to that?
It's easy to damage, isn't it?
Right, you know, if someone hits you in the head, that's bad. So we do have a skull, yes, which is good, but you know, if somebody whacks you in the head, they take the whole organism out. If somebody hits you in the arm or the knee, well, that hurts, but the rest of you is still functional. If you get hit in the head, it's a problem. So yeah, you have to worry about damage. But in general, cephalization is evolutionarily advantageous. So that's why we have a brain in our head. Of course, if you were like my parents, sometimes they may question that, right? Do you have a brain in your head? So there you go. So yeah, close-domain sensory organs, so faster information transfer, identify things sooner if we lead with our head. Okay, so your nervous system is composed of basically two types of cells. And the main cell that we're interested in is the nerve cell, also called the neuron. So neurons have a very characteristic structure. It's really easy to look at a piece of tissue under a microscope and tell if a cell is a neuron or not. Most cells are just kind of round-ish, right? Neurons have these very, very characteristic processes. So when you look at a neural cell, there are really four components that you can identify that mark it as a neural cell. So the first thing is, on one side of the cell has all these little protrusions that are called dendrites. It looks like a little tree, and in fact, it's called a dendritic arbor, which is just like a tree, right? Arbor Day. Then you have a cell body, right? All cells have a nucleus, they have organelles, they have stuff to keep them alive. That's the cell body of the neuron. Then most neurons have a single, very long process called an axon. And the axon enables this nerve cell to connect with other nerve cells. Okay. Sometimes axons are very short, like in your brain. Sometimes axons can be only a few millionths of a meter long. So 1/1,000,000th, the prefix is micro. So some axons are only a few micrometers in length. And the little micro is a mu. They can be very short or they can be really long. So for example, the nerves that move your big toe, flex your big toe right now. The cell body of those nerves is in your lumbar spinal cord. The axon goes all the way down your leg to your toe. So depending on how tall you are, those axons can be a meter or more, greater than 1 meter. And if you're a giraffe, they could probably be 2 or 3 meters long. So axons can be really long. So axon's the third part. And then the fourth part of a neuron is the synapse. The synapse is basically where one neuron connects to another one. One thing you'll notice about the synapse, and we'll talk about this in more detail later, is that it's a physical gap. The neurons never actually touch. There's always a tiny, tiny bit of space between one neuron and the next. In terms of terminology, when we look at two neurons that are joined by a synapse, the one where information flows this way, information flows from the dendrites down the axon to the synapse. The one through which information is passing is the presynaptic cell. The one that is receiving the information is called the postsynaptic cell. And oftentimes what will happen is the information from the presynaptic cell comes into the postsynaptic cell, and then it passes that information on to the next cells in line. So it's like communication, it's like a line of communication, right? And this is an actual image of a neuron. This is a particular type of neuron called a pyramidal cell. The reason is because the cell body kind of looks like a triangle. But you can see lots of dendrites, right? The dendritic arbor. You can see the axon. And in this case, the axon splits up into a few different little fibers. And then down here at the end, you can't see, it's too small, are the synapses. So, dendrites, cell body, axon, synapse. 4 main components of a neuron. That's the way the information flows. It comes in through the dendrites, goes out through the axon to the synapse. So, these are important, so you should know that. So in fact, why don't you go right now, go on your iPad, just find a little space on Notability or whatever you're doing, and draw a neuron. Draw a neuron for me, just from memory. Doesn't have to be pretty, but make sure you include the 4 main components. conducting. Okay. So I'll let you guys wrap it up. Doesn't have to be, Rembrandt or anything, right? So, a neuron, right? Information flows this way, from left to right. When I mean information, what I mean is it's a nervous impulse, but it means something. So for example, it could be, hey, this hurts, right? You stick your finger with a pin, the pain information is going to go all the way up to your brain. Your brain's going to interpret that as, hey, this hurts. It's going to send a signal down to your arm to say, move your finger away from that, whatever it is, it's bad. So information could be pain, it could be hunger, it could be anything. Nerves pass those on from one to another. So in terms of a single neuron, where does the information come in? What's the first component of the neuron, right? We talked about the 4 components. What's the first one?
Dendrites.
The dendrites, exactly. So, right, all these nice little trees, right? Just a nice way to collect large amounts of information, right? If you want to listen to lots of noises, you're going to use a parabolic microphone, which collects a lot of information. Dendrites do the same thing, spread out over a wide area, collect a lot of information. What are the dendrites connected to?
The cell body.
The cell body, also called the soma. Right, so information flows into the dendrites, into the soma. What's the next part?
The axon.
The axon, yes, right? Generally, a single long process can be very, very long, which carries the information further until the axon ends in what? The synapse, yeah, the synaptic terminals. Where that information is passed on to another neuron. So this is a very stereotypical, not very well drawn, but if you look at a cell and you see all this complexity, it's a neuron. It's very clear. It's impossible to mistake a neuron for any other type of cell. But know those 4 parts: dendrites, cell body or soma, axon, synapse, and know which direction information flows. From the dendrites to the synapse. Good? So I already kind of had myself a little bit talking about how long your axons can be. Right, so like a giraffe, right, their axon is 2 meters or more. So there is a point of diminishing returns. The longer the axon is, right, just like anything, any communication, distance is a problem. The further away you are, the less communication there is, right? That's why long distance relationships don't work. The further apart you are, the less information is passed. So having a super long axon is not as good as having a short one. So there are limits, but we know that those can be at least several meters in mammals and vertebrates. So this cell, this super long neuron, right, which has a 2 meter long axon, what might you think as a potential problem for that cell? A cell with a super long axon. Kind of already talked about one thing, right? Communication is a little bit slower. What's another thing? Taylor?
So it's like very long, it's like takes the entire length of the leg, so like if that nerve gets damaged, then like the whole leg would be unable to function?
Yes, so nerves are actually bundles of axons.
Yeah.
Nerves are made up of hundreds, sometimes thousands of axons. So you could partly damage a nerve
Yeah.
where some of the axons are cut. But yeah, if you completely destroy,
If only like
completely
destroy
sever.
that long nerve, then yeah.
Yeah, but the longer it is, right, the more likely that can happen.
Yeah.
Right, so, yeah, so the potential to damage the nerve. There's one other thing, because, right, the soma has the nucleus and all the machinery to make proteins and stuff like that. But if they're all made in the cell body, they have to get transported physically down the axon to the synapse. And the longer they have to be transported, the longer it's going to take. So it's hard for neurons like that to repair themselves, and also harder for them to keep the synapse supplied with all the biochemical machinery it needs to function. So there is a problem with, called supply, right? The supply chain. So the longer neurons, the larger neurons get, the longer axons get, there are problems that come up. But, right, it works. It clearly works, right? You can argue about it all day, but the giraffe clearly can walk with a 2-meter axon down its leg. So it works. It may not be optimal, but evolution doesn't care about optimal. Evolution doesn't care about perfection, right? Good enough. As long as this thing can get around and eat and reproduce and pass on its genes, evolution is like, cool. Okay, my job here is done. Okay, any questions? All right, so the nervous system is divided up into 2 main parts: the peripheral nervous system and the central nervous system. Central nervous system is basically your brain and spinal cord. The peripheral nervous system is everything else. In order for us to function, we have basically 2 types of nerves. We have nerves that gather sensory input, and sensory input could be what? What are some types of sensory input?
Just sound.
Vision, right? Smell, taste, touch. Those are all sensory inputs. That's how we survive negotiating the world around us. We take sensory inputs. You know, if we hear a big truck coming, we know it's not a good time to dart out to the street. So our sensory inputs, those come in through what are called sensory neurons. Sensory neurons are part of the peripheral nervous system, and they carry information from the environment to the brain. The brain is composed of interneurons. Those are neurons that connect to each other, and they connect a sensory input to a motor output. Motor output just means moving a muscle. Okay? So sensory input in the peripheral nervous system, integration in the central nervous system, motor output in the peripheral nervous system. The thing that takes in the sensory input, whether it's your ear, your eye, is called a sensor. The muscle that makes the motor output is called the effector. It makes things, makes things happen, it affects things. So you can kind of put this all into somebody hitting a baseball, right? So the hitter has all sorts of sensory input. They can see the pitcher, they can see when the pitcher releases the ball, they know the distance between the mound and home plate. So they take all this sensory information in. The brain says, okay, this appears to be a curveball. If you want to try to hit it, you need to start moving your bat at point X. You need to make sure your bat is right in the middle of the plate when the ball gets there so you can hit it, and make sure you follow through. And all of that information then gets turned into motor output, making all the muscles in your arms, your legs, your body cooperate to bring the bat to a particular place at a particular time. And if you're lucky, right, you hit the ball, you connect, and it goes out the park from your home, you hit a home run. All of that happens in less than a second. So that's another thing about the nervous system, is it's incredibly fast, right? There has yet to be a computer designed with fast enough processors to hit a baseball like a human being does. And yet, we do this all the time, right? Even if you're not a baseball player, you probably played baseball as a kid, you did all of this stuff. And that was bang, all this processing, all this thinking in 1 second or less. So the nervous system is incredibly fast. And because we have big brains, incredibly complex things can happen very quickly, like hitting a baseball. So almost everything we do requires a sensory input, motor integration, or integration and then motor output, right? It doesn't have to be external stimuli either. You can get some sensory input from within your body. Let's say that your stomach is empty, it's shrinking. There are actually neurons in your stomach that feel that shrink, and they send messages to the brain saying, hey, your stomach's empty, maybe you ought to go get something to eat. Your brain thinks about this, looks at the clock, says, okay, the dining hall's open, it's 11:30, and then the motor output is to walk your butt down the hill to the dining hall to get something to eat. Same thing as hitting a baseball. Not as fast, but you've got an input, integration, and an output. So basically any action that you do, you can break it down into those three components. Right? Sensory information, integration, output. Sensory neurons carry the sensory input. Motor neurons of the brain do this thinking, and motor neurons do the output. So those three types of neurons are the main types of neurons in the nervous system. Okay, so I said neurons were one of the two main types. The other type of cells are called glia. So the two main types of cells are neurons and glia. Right. Neurons do all the work, right? They do all the thinking, sensing, motor outputs, everything else. The glial cells are there to support the neurons, to help them out. They have a support role. Right? Just like any good athlete needs a coach and an agent and right, entourage or whatever. Every big star needs an entourage, right? The star is the neuron, the entourage is the glia. What's one thing we know about stars versus entourages? Which is bigger? Right? You know, one star, you've got a big entourage. Bigger star, a bigger entourage. Same thing is true with glia. You have about 100 billion neurons in your brain. You have over trillion glia. So there are more helping cells than there are neurons, because neurons are actually pretty wimpy when you get right down to it. When a cell becomes a neuron, it's what's called terminally differentiated. It will never ever divide again. And so that's what people say when you do damage to a nerve cell, it's generally not going to repair itself. It'll die, because neurons are pretty wimpy. So glial cells try to help out and keep the neurons as healthy and happy as possible. Now, we are learning more about neurons. We are learning that they do have the ability to repair themselves to some extent. In some animals, neurons can regenerate, the axons can regenerate themselves. We know, for example, in the axolotl. Anyone have an axolotl?
My friend does.
They're little cute little guys. If you sever a limb on an axolotl, it'll regrow, and the severed nerve will regrow and be functional. Humans, we can't do that. For whatever reason, our neurons cannot regenerate.
In the most extreme cases with axolotls, you can sever the head of a second axolotl and stitch it onto a first, and both will survive and both will control the body.
Yeah, up to a certain point, though. I think. I don't think
That's
you
like
can-
the extreme
Yeah.
extreme with that, but both will still survive.
You can do a lot with like embryonic amphibians, like axolotls or frogs. So yeah, so like a tadpole. When I was a graduate student, I worked with frogs and frog embryos, and you can take one half of a frog embryo and stick it together with the head with the tail, and it will turn into a frog. Can't do that with human embryos. Don't even try. You'll go to jail. But yeah, so nerves, especially in mammals, are not really very strong. So there are a couple of different types of glial cells. Actually, there are more than this, but these are just the ones I'm going to mention. So astrocytes are glial cells that help to provide metabolites, food, to the cell. Neurons are pretty wimpy. They like to get their food basically already kind of pre-digested, pre-chewed up. So that's what astrocytes do. Astrocytes are the component of what's called the blood-brain barrier. So astrocytes have these little processes that reach out to the capillaries that pass through your brain. They take in the food and the nutrients and the oxygen that your body is supplying, and then they carry that to the neuron and give it to the neuron. So, right, it's kind of like the king, right, has a poison checker and eat everything before the king eats it. Because bad stuff, if it gets through the astrocyte, can kill the neuron. So that's why, like, the blood-brain barrier is important. Why astrocytes are important. If the blood-brain barrier is ever broken for some reason, then things can get straight into the neurons, and the neurons are pretty helpless. The other types of glia are what are called myelinating glia. Myelin is a very interesting protein. So only vertebrates have myelin. Invertebrates, insects, echinoderms don't. Myelin is a protein that the myelinating glia make. And these surround the axon. So in the peripheral nervous system, the myelinated glial cells are called Schwann cells. And they wrap themselves around the axon, kind of like a tortilla around a burrito. In the central nervous system, just to make it complicated, they're called oligodendrocytes. They're slightly different, but they do the same thing. Yes. Well, Schwann got the cell named after him. Oligodendrocytes can actually myelinate multiple neurons. So when you look at them under a microscope, they look like they have lots of branches. Dendrite.
Yeah.
Oligo means multiple.
Okay.
So it's, oh, here's a cell with multiple branches. I'll call it an oligodendrocyte. Most people name things before they find out what they do, which sometimes leads to pretty stupid names for things because we figure out what they do later and it's like, oh, yeah, okay. But yeah, so sometimes in science, the name doesn't make much sense because they named it before they figured out what it did. Right? Everybody wants to get their naming right. Schwann wanted to get his name in. I want a cell named after him. Well, at least, people 100 years after he died still know his name because he named those cells. So myelinating glia, oligodendrocytes and Schwann cells, wrap around the axon. Now, if you know anything about electronics, right, you know that when you connect two things with a wire, you have to have bare ends. But what's the middle of the wire covered with?
Insulation?
Insulation, yeah, like a little plastic sheet, right? So electricity doesn't go out all over the wire and shock you, just at the ends. Myelin is basically insulation for the axon. And it accomplishes the same purpose, basically. The energy goes from one end to the other without much of a loss in the middle because the axons are wrapped with these myelinating glia. So myelin helps information move from cell body to synapse with minimal loss of information. So we can say a stronger signal and also a faster signal. Myelination speeds up how quickly a signal gets from here to here. And actually, myelin is really an interesting topic. This is really all we're going to do, but I could teach a whole course on myelin. So only vertebrates have myelin. Only vertebrates have big brains with lots of neurons in it. One thing, if you have a big brain, right, is you want the impulses to be fast. The faster they are, the faster you can think, the more quickly you can react to a threat, the longer you stay healthy and reproduce and pass these things on to your children. So myelinated axons can be very, very thin, much, much thinner than a human hair. So a myelinated axon, if you chop that look in cross-section, would look like this. Here's the axon, here's the myelin sheath around it. And information passes very fast down a myelinated axon. If you do not possess a myelinated nervous system, like a spider or a crab, in order to get an equivalent speed, your axon has to be this big. Right? Because you can push a lot of information through this little hole. If you have a bigger pipe, you can push more information through. But insects, invertebrates, have much larger axons. if you have a larger axon, you can't fit as many neurons in your brain. And so the brains of insects are significantly smaller because they can only have a few neurons in them because the axons are so thick. Myelinated axons mean you can have very thin axons, you can put a lot of them in the brain, you can make a lot of connections. That's how mammals, that's how vertebrates evolved with myelin. We can make very, very compact brains with lots and lots and lots of connections. So, if you ever go watch monster movies, do not worry, spiders and ants will never take over the world. And the reason is because if they were to have a brain that was as complex as ours, their heads would be like larger than this building. And so they would all flop over from the weight. So invertebrates will never take over the world because they don't have myelin. So there you go. All right. So we're going to start this is a little bit complex part of the lecture. And I'm going to kind of touch on it now, and then I'm going to go over it again on Friday when we start up. So I mentioned on Monday, right, that when we talk about information, in neurons, information is carried basically by an electrical current, the movement of charged ions. So it's a change in charge, right? You can basically have three types of charge. You can have positive charge, right? You can have a negative charge, and you can have one that is neutral, not charged. And anytime you change from positive to negative, you create an electrical current. And so your neurons do that by moving positively and negatively charged ions across their cell membrane. That changes the polarity of the membrane, and it moves basically in a wave of polarity change from the cell soma down to the synapse. So this is kind of a picture of a tiny, tiny piece of axon. Okay, so let's imagine we take a tiny, tiny little piece of the axon membrane out, and we blow it up to this huge thing. Right, that's what's on the board. I'm not going to try to draw this because I'm really not good at art. So the cell membrane is made up of what? You guys remember? I'm sure you probably did cell membranes in high school biology.
Phospholipids.
It's phospholipids. It's called a lipid bilayer. So basically it's two layers of phospholipids, one with the heads pointing out, this is the outside world, and one with the heads pointing into the cell cytoplasm. In general, I don't know why it is, but in almost all of these diagrams, the outside world is nice and sky blue, and the inside is kind of a pinkish beige color. I don't know why they do. Every textbook looks exactly the same. So electrical charged particles cannot pass directly through the lipid bilayer. They are prevented. So the cell has evolved what are called ion channels. These are protein channels within the membrane, literal holes that allow the passage of charged ions. And when we're talking about a nerve, and we're talking about electrical impulses in a nerve cell, there are two main ions that we're concerned about. One of them is sodium, and I think sodium is, the little yellow blobs. And one of them is potassium, which are the little orangey diamond-shaped things. So the two main ions that are responsible for communication, for signals moving through the nervous system, are sodium and potassium. So in the cell membrane, there are two types of ion channels. One only allows potassium through, and one only allows sodium. So they're very specific. in general, a cell at rest, when it's not doing anything, has a net negative charge inside. That's because of things like DNA and proteins, which are stuck inside the cell, which never leave, all have an overall negative charge. So inside the cell, all the cytoplasm inside the cell has a negative charge relative to the outside. Right? This is all relative. So the inside of the cell is more negative than the outside. In order to pass an electrical impulse, that charge difference has to shift. So the membrane has to go from positive outside to positive inside, and negative inside to negative outside. And that change is accomplished by allowing these ions to pass through the ion channels. So you guys remember the process of diffusion? You might have done that in high school chemistry, but you observe it every day, right? So simple diffusion, when I say something diffuses, what do I mean? When you put a drop of dye in a glass of water, right?
Like it spreads out?
It spreads out. Diffusion, substances move from an area of higher concentration to an area of lower concentration. It's a fundamental physical property of this world, right? Substances move from an area of higher concentration to an area of lower concentration. So that's why your drop of colored dye spreads out in the water until, right, the entire water is colored and it reaches what? What's it called when two systems are balanced?
Equilibrium?
Equilibrium, right? It reaches equilibrium. But until then, things move from higher concentration to lower concentration. That's why if someone walks into, walks into a room and they're wearing a strong perfume or something like that, the first people that are going to smell it are the people in the front. It's going to take a while for those odor molecules to diffuse all the way to the back. Eventually, all the odor molecules will be all over and everybody will smell it. But diffusion starts at the highest concentration and goes to the lowest concentration. Your neurons rely on that to move these charged particles across the membrane. So in a resting neuron, the outside has much more sodium than the inside, and the inside has much more potassium than the outside. This is a disequilibrium. In order to achieve that as a living organism, you have to apply energy, right? Because otherwise, everything comes to equilibrium. Second law of thermodynamics, right? As a living organism, you do not want to come to equilibrium with your environment, because if you do, that means you're dead. So your cell has to maintain this gradient. And how it does it is using this guy. This is called the sodium-potassium pump, or sodium-potassium ATPase. So it is active 24/7, 365. And what it does is it pumps 3 sodium ions out of the cell in exchange for 2 potassium ions pumped into the cell. And because these are moving against their concentration gradient, they cannot diffuse, it requires energy to move them. Just like water flows downhill, but you need a pump to move it uphill. Things diffuse from higher to lower concentration by themselves, but if you want to move something from a lower to a higher concentration, you need energy. This guy uses cellular energy in the form of what? What molecule is associated with cellular energy?
ATP?
ATP, exactly. The sodium potassium pump is using ATP 24/7, 365. So remember on Monday I said, your brain mass is only 2% of your body weight, but it uses 20% of your energy. Why? This guy. This guy is frantically pumping sodium out and potassium in every neuron in your brain all the time. So it creates this disequilibrium. Now this is the normal, the normal environment for a nerve cell. So when an electrical impulse comes along, these ion channels open, sodium rushes in, and sodium is positively charged. So sodium carries a positive charge inside, and the inside becomes more positive. And then, this is in sequence, the potassium channel is open, potassium is positively charged, and it leaves the cell. When positive charge leaves, the inside of the cell becomes more negative. So you get this very brief change of the cell polarity from positive inside, or positive outside to negative outside. So if we think about an axon moving information, right, it basically moves by this change of membrane potential, this temporary change of membrane potential going down the axon. Yes?
What would happen if the outside and inside of the cell are equal? Because if it has an equilibrium, like if the charges like on the outside and inside are kind of like equal, you're not going from high concentration to low anymore. Would
You'd be
they be
dead.
able to come into the cell anymore, or?
So,
that's
yeah,
not
actually,
how it
I was gonna get to that, but right, once things reach equilibrium, then they're equal concentrations on either side. Now, does that mean that once that happens, nothing moves? No, everything's always moving. At the microscopic level, right, we have what's called Brownian motion. All of these atoms and molecules are vibrating at incredibly high frequency. They're all bouncing around inside your cells. But when something reaches equilibrium, all it means is that there's no net change in the concentration. So for example, if we get to equilibrium where the amount of sodium on this side is equal to the amount of this side, then for every sodium ion that passes this way, one has to pass that way. Okay. But generally, once you're at equilibrium, you can't do anything. You can't do any work. You can't send a nervous signal. Equilibrium in a living thing is generally pretty bad. So the major players in a nervous impulse are ion channels and pumps, the two ions, and the change in charge. So those are four components that you should know. All right. So this is how you can actually detect that. So the sodium potassium ATPase, sodium ions and potassium ions, and sodium channels and potassium channels. Those are the major components of the cell membrane that allows charge across the membrane to change. So this was all kind of sussed out about 100 years ago by the earliest neuroscientists. And what they learned was you could put a microscopic electrode inside an axon. And they used invertebrates for this. Why?
No myelin.
No myelin, and so they have giant axons. They're much easier to stick an electrode into than a tiny, tiny little burger one, which is a good thing, because otherwise we never would have figured this stuff out. So when you do, you can measure the electrical potential inside the cell, and you can compare it to a reference electrode, which is outside the cell. So say you have a cell in a little saline dish back. You can compare the charge outside to the charge inside. When you do that to a neuron that is at rest, it's not sending any information, it's not doing anything. The inside is more negative than the outside by about 65 millivolts. So the electrical gradient across the membrane in a resting cell is minus 65 millivolts, which means it's more negative inside than outside. A millivolt is 1,000th of a volt, right? So ever touch a 9-volt battery to your tongue, that little zap? So that's 9 volts. This is 10,000 times smaller. So it's a really, really tiny amount of electricity. But if a bunch of neurons are working together, they create enough electricity that if you put an electrode on your skin, it can pick up that electrical activity. And that's how you do things like do electroencephalograms, where you can read people's, or you can look at people's brain waves. Those are actually movement of electrical charge across a cell membrane. Okay. Now, when a cell is more negative inside than outside, it is said to be hyperpolarized. That means that there's a big difference between the inside and the outside. When something is polarized, right, people talk about, right, our country is polarized because there's a big difference between one side and the other. Same thing with a neuron. They're hyperpolarized. There's a big difference between here and here. In the case of a resting neuron, sodium is high outside. You know, see, they reverse this one, right? So usually outside is on the top, and this one they put inside on the top. So sodium is higher outside, lower inside. Potassium is higher inside and lower outside. And the sodium potassium pump, which is kind of in the picture here, is constantly moving potassium in and sodium out. These are the sodium potassium channels. Okay. One thing to know is these channels are what are called gated. And by gated, they just basically have a gate. They can be either open or closed. If they're open, the ions can freely pass through. If they're closed, they can't. So ion channels, ones that are called gated, are basically binary, just like a logic gate. They're either open, well, it's not entirely true. Okay. They're not entirely binary. There's open and closed. That's as far as we'll go for today, because it's going to get more confusing, if it's not confusing now. So, when a nerve cell membrane is at rest, this is what it looks like. Sodium is high outside, potassium is high inside, the sodium potassium pump is constantly moving things, and the ion channels, the gated ion channels are all closed. So at this point, no ion can cross the membrane, right? Can't go through the channels, can't go through the membrane. This is what your nerve looks like when it's resting. It's not doing anything, carrying information when you're sleeping. Even though when you're asleep, there's still information. We'll talk about it at rest. So a nerve cell at rest is said to be hyperpolarized. And in general, it's minus 65 millivolts. All right, yeah. Okay, so resting potential. So minus 65 millivolts is what's called the resting potential. So how do we model the resting potential? So this is kind of an artificial example, right? This is a beaker with a membrane down the middle which has potassium channels in it. And it has a bunch of potassium on one side that is very concentrated, 140 millimoles. And on the other side is very dilute, only 5 millimoles. So we've set up a concentration gradient here across this membrane. Remember, these guys can't cross this membrane, they can only go through here. So what happens if all of these guys are closed? Right, nothing moves across, right? Now, if I open these, which direction would the potassium flow? Left to right, exactly, right? It would go down its concentration gradient. So basically, when you open ion channels, the concentration gradient will push the stuff at higher concentration across the membrane. Because these are charged, right, that's like moving an electrical charge. So a positive charge moves from here to here. So that's what the resting potential of a neuron, this is how you can kind of model it. It's as a barrier between two chambers that have different concentrations of ions and whether the channels are open or closed. If they're closed, then nothing moves across the membrane and this difference exists, right? It's hyperpolarized. Once these ion channels open, positive charge moves in and the difference is going to start to change. And this works for sodium as well, except in reverse. Now, one thing somebody asked, right, is, well, what happens if the potassium channels are open until everything's at equilibrium? What kind of charge difference do we get then? So in order to, we can model that mathematically by using the Nernst equation. So you can predict the equilibrium potential of a neuron, that is when the chemical concentration and the electrical concentration are exactly balanced, using the Nernst equation. Okay, because there are two forces here at play. One is concentration. So in that example, potassium is moving down its concentration gradient from right to left. So we can model a potassium gradient like this. It's big on the left side, and it gets smaller to the right. Generally, when you enclose something with brackets like this, it means concentration, the actual number of molecules. Okay? So in general, potassium will go from left to right. However, there is a second component. So concentration is one, electrical charge is the other one. Right? Because what do we know about like charges? What do like charges do? They repel each other. Exactly. So as the charge gradient, as potassium moves down its concentration gradient, then more positive charge is moving this way. And since potassium is positive, the amount of energy pushing back on the potassium is increasing. So while the concentration gradient goes left to right, the electrical gradient goes right to left. The more potassium that flows across the gradient, it's moving down its concentration gradient, but it's moving from an area that has less positive charge to an area that has more positive charge. So that's going to start pushing back. When the system is at equilibrium is when these two gradients are balanced. The concentration gradient, the force pushing left to right, is exactly balanced with the charge gradient, the force pushing right to left, somewhere in the middle. And the Nernst equation allows us to calculate that. But don't panic. I'm not going to ask you to calculate anything using the Nernst equation. You don't have to memorize it. If I want you to use it on a test, I'll give it to you. Okay? No point in memorizing this. You can find it anywhere. But what Nernst equation says is basically there are two components to the equilibrium potential. Well, three actually. The first one is the ion itself. So equilibrium potential is specific to each ion. So you calculate a potassium equilibrium, you calculate a sodium equilibrium. They're going to be different. There's a constant in the Nernst equation. In this case, it's 62 millivolts. Basically, this takes into account everything in the system except the concentration. So, for example, right, if something's moving through Jell-O, it goes a lot slower than if it's moving through water. So the viscosity of the medium affects the equilibrium potential. So since we're talking about biological systems, always assume that biological systems are just in water at room temperature. It's not always true, but it makes calculations all easier. But that constant takes things like that into account. So just know that that number means something about the overall system. And it's different from cell to cell, it's different from organism to organism, but this works for mammalian neurons in culture. And then this is just the log of the concentration difference, right? 5 millimolar versus 140 millimolar, right? So you take the log of 5 over 100, or 140, sorry. Right, that gives you negative 90 millivolts. Now let's say that I change the concentration on both sides. Let's say the concentration on both sides is now equal. Right, so remember we have the 62 here, multiplied by this, equals minus 90. Let's say the two concentrations are equal. So you have 75 on one side, 75 on the other. This fraction is 1. What's that going to do to the number? Well, you don't have to do the calculations, but basically, right, it's going to reduce, it's going to make this number smaller. Well, it's going to be less than minus 90 anyway. I can't do it in my head. So basically, by changing the concentrations on both sides, you change the equilibrium potential. That's all you need to know about the Nernst equation. Nernst equation tells you that whatever the concentration of the ion is, if you change it, you will affect the equilibrium potential. It'll either go up or down, depending on if this is more highly polarized. So let's say we've got 1 millimolar versus 1,000, right? Then the potential is going to be huge across the membrane. If we have it at equilibrium, it's going to be, this is going to be essentially 0, so you're going to just get the constant, so 62 millivolts. So, but minus 90 millivolts is called the equilibrium potential for potassium. That is, if these are left open, potassium will come to an equilibrium with a difference of about 90 millivolts. And it'll be more negative on the outside than the inside. That's where the electrochemical and the concentration gradients equalize for potassium, minus 90 millivolts. So basically, if you left if the neuron left its doors open, right, the neuron just left the ion channel doors open and potassium was free to equilibrate, ultimately the charge across the cell membrane would be minus 90. It never gets that way unless the cell's dead. In fact, it never gets that way because then it will just consider all done. So the Nernst equation can tell you something about when you change the concentration difference between the sides, you change the equilibrium potential. And that's going to be important next time when we talk about action potentials, which are literally the movement of these changes in charge down the length of the membrane, which is what carries the information from the cell body to the synapse. Okay? So I know this is a complex concept. I will go over it again Friday, so you kind of